All right, here's our video today. How amino acids make proteins. And you can see them in the background there. They're not really gumballs. They don't look like gumballs, but just for teaching purposes. So you got lysine and glycine and so forth. And we'll talk about that in the videos. This is very important to know, though. Without amino acids, you just wouldn't exist. Period. Simple as that. You need amino acids to make proteins. And proteins, very important. Proteins are needed for virtually every biological function. So you just wouldn't exist without proteins. And there's 500 amino acids in nature, but for us, there's 20 in us. And well, there's two other special amino acids that could be made. And so out of that 20, 9 we don't make. The body doesn't make those 9, so we have to get them from food. So basically, we're looking at 20 amino acids that could make an infinite amount of proteins. We'll talk about that in the video. And here's a little analogy that'll help you. Think of amino acids as letters in the alphabet, A, B, C, and so forth, in the English alphabet or any alphabet, and proteins as the words. In fact, if we go further, because from amino acids you can make peptides, and you can make proteins from those peptides. So think of amino acids as the alphabet. They're the letters, and the words will be the peptides or sentences, and the proteins will be books. That's how we can think of it. So let's write that analogy. I'll use yellow. That's what I'll use. So again, when we think of amino acids, the building blocks for proteins, think of them as words. Well, not words. We should think of them as letters. Letters, not words. And we think of the peptides, and we'll look at peptide chains. We'll think of them as the words. And for proteins that eventually they'll become, we'll think of them as the book. So that's the analogy we're going to be using. So how are they made? That's what this video is about. How are they made? So we'll think of the word you'll hear is protein synthesis. So I'll write it out. It's a fancy word. And just synthesis is just making, the building of protein. So protein synthesis, and it happens first, we'll talk about in transcription. And that word transcribe, think of that word transcribing. And we'll get to an analogy on that. So first we're going to build proteins. We're going to do transcription first, and it's going to happen in the nucleus. It's going to happen in the DNA, which is in the nucleus. In the nucleus, and then what happened to that letter there? Let's write that better. Put the C and the L together, we made a D. We had a mutation. So in the nucleus, and we're talking about DNA, deoxyribonucleic acid, or the nickname the double helix. So this is where we're going to have an mRNA. It's going to transcribe what's in your DNA. It's going to leave the DNA. It's going to find a ribosome, and it's going to make proteins. Simple as that, and we'll explain the detail of it. But first, in the cell, in the nucleuses, you have your DNA, and it's long and it has genes on it, and it has to be unwound. And there's this enzyme that will break it down. It's a protein, which are enzymes. Enzymes are proteins, and it's called a helicase, and it's going to unwind, or it's going to break the hydrogen bonds in DNA. So let's look at that. So here's your double helix, the DNA. And we have our bases, these genes on the DNA. Let's make sure our pen's working. So the bases, there's only 4 bases in DNA. For example, it'll be adenine. Make sure the little thicker A there. Adenine, there we know. There's adenine, and which will always bond with thymine, and vice versa. And then there's guanine, which will bond with cytosine. And so if you had, for example, A, G, 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 C, T, for example, well, What will that bond with? Well, the A will bond with thymine, T. The G with C, G with C, G with C, C with G, and T with the A in the DNA. And it'll be different when mRNA comes around because they will not use thymine, which we'll talk about that. So here's the helicase right here. Write down the helicase, which is a protein. And here's something weird, right? You need proteins to make proteins, just like you need energy to make energy in the body. Very important. I'll say it again. We need proteins to make proteins. And this helicase right here, it's breaking the hydrogen bonds because these nucleotide bases that I mentioned, adenine and thymine and guanine and cytosine, they have, and we'll use yellow, they have this hydrogen bond that bonds them. Actually, there's 3 bonds in between guanine and cytosine and 2 for adenine and thymine. So here it is, the helicase is breaking it apart so a RNA molecule called the messenger RNA could code, could transcribe. So let's just make a little RNA here, DNA that is. Well, it's going back to this one. Well, we'll keep it here. So we'll draw a mRNA molecule that is formed, and it will come over here, and it will start coding by the bases. All right, let's try to clear that up. So let's just say on top here we have our DNA that's unwound, and over here we'll just put some bases. Let's say we have an A and a T and a C and a G and another G and another A, let's say. So there will be a complementary strand. This is called the sense strand. This is not important right away, but it's the sense strand. This is the anti-sense strand. So as it codes, a complementary here will be thymine. This will be adenine. And I'll just put the complements in here. G with C always. You can remember it by car in the garage or a tree, A-T, a tree, or whatever other mnemonic device you want to use. So here it is. These are the complementary strand in the DNA. And when RNA comes over, let's say RNA over here, this RNA molecule, it's going to code in the antisense strand. So this would be, instead of using a thymine, like I said before, they're going to use uracil. So A will become U, T will become A, C will become G, G will become C, and T. So now we have this is the mRNA strand. It has transcribed it. Now we can think of transcribing, think of a monk or a scribe from the Middle Ages and so forth. They copy the book as it is. They're copying the book. And there's a little errors here. Instead of putting a T, they put a U. And look, with any transcriptions, there could be some errors involved. So once the mRNA transcribes it, and we're thinking of, I'll just put the analogy here, a scribe, transcribe, copy, scribe will copy the book and your genes. It's copying your instructions. Just like scribes had to do this before the printing press, before Johannes Gutenberg introduced the printing press in 1440, we had scribes copying the books, word for word. Okay, once it's copied, the mRNA molecule is going to leave the nucleus and it's going to look for a ribosome. And in the ribosome that's where amino acids will be made, where peptides will be made, and that's translation, which we'll talk about. And here we have the mRNA left the nucleus. It's going to go into a ribosome. Transcription is over. Now we're going to think of translation. And if we keep that analogy with translation now, we have the book translated. We copied the book. We haven't translated it yet, though. It's just copied. Now we want to translate it into another language. So that's basically what's happening in translation. The book is copied. Your instructions are copied in the nucleus from the mRNA. And now it's going to go find the ribosome, and with the help of tRNA, they're going to translate the book. They're going to copy it in another language. All right, and here's another picture of it. The mRNA leaves the nucleus. It's entering the cytoplasm where the ribosomes are, and here it is entering the ribosome. It will enter it in codes of in threes, and those threes are called codons. So let's write codon down there. So we'll write codons. So codons are just three bases. They're coming in three at a time. And the tRNA is going to match it. See here, this three over here, AAA, it has to be matched from the tRNA, and they're going to bring in UUU. Because remember, we don't use thymine anymore. We're using uracil. So the tRNA will bring this UUU, by the way, which is lysine, and they'll bring it in as it enters the ribosome. So let's make sure we understand that codons are groups of three. Now, before this could happen, they need to know, and this is what's pretty amazing about the body, is they need to know when to start and when to stop. And you can see on the bottom of this picture over here, there's a start codon. So it needs this codon to start, or else it won't start. And then while it's coding, while it's translating, it needs to know when to stop. It's like someone saying it's quitting time, or someone saying, oh yeah, that's all. The editor is saying, that's it. That's all we want to put in this book. So it needs to know when to start, and it needs to know when to stop. So there'll be a start codon, and then somewhere down the line, there'll be a stop codon, which will tell it stop coding, which is amazing. So just to review, to make sure we understand this, we know that mRNA is going to copy your DNA. It's going to transcribe it. Think of scribing the book, and then think of the word late as the next step. It's late, it's later. Then it will translate. So it's copying the book in the nucleus, it goes to the ribosome, and there's going to be these special translators that will translate the book into another language. In other words, taking your DNA and now making amino acids as another language in the analogy. By the way, here's the stop codon, UAA. It's a stop codon. And by the way, as you see the tRNA with the UUU, that's called the anticodon. It's the three bases that are complementary to the one that's trying to enter the ribosome. And an explanation of that, if you want to understand why the body is not using, why thymine doesn't leave the nucleus. Remember, thymine does not leave the nucleus. Thymine is, in a way, you can say it's more economically cost effective to use uracil, because mRNA doesn't last long, so you don't really need it for durability. But you need thymine. Thymine, you need it for durability and structure, so that's why it's in DNA. But RNA can get away with using uracil because it's almost like your thymine, and it's more economically cost effective if you're thinking about economics in an analogy. We're not using money, but we're talking about in energy. So, in other words, uracil is more flexible, and you don't need additional enzymes. That's why it's more cost effective. So anyway, just to take away from this, uracil is replacing thymine outside the body. So now we have these codons, which is three pairs at a time coming in, and amino acids are being created in the ribosome. Here's another picture of it. So as you can see, as it's coming in, make sure my pen's ready. As it comes in, these amino acids are being made. So you can see the amino acids are being made here. And eventually you'll have a chain of them. Let's get a better color. You'll get a better chain of amino acids forming, and this will be a peptide chain. So you have amino acids forming a peptide chain. Now, once that peptide chain is made, once there's enough of it, and once you have a stop codon, then there's another protein that's going to help out. They're called chaperone proteins, and they help fold the peptides to make proteins, and they'll be in the cytoplasm. So here's a little crude picture of the chaperone. So the polypeptides, these peptide chains of amino acids, they enter into the chaperone and they help fold it. They're just folding it, making it ready for packaging. So that's what happens after they leave the ribosome. So again, just a review how amino acids make proteins. Amino acids in the transcription part, the mRNA will transcribe, they'll code, they'll copy the book. That's the analogy. They'll leave the nucleus, find a ribosome. They will go in groups of 3 bases. Those are codons with a complementary anticodon from the tRNA. And as they enter the ribosome, amino acids are formed. Those amino acids will make peptides, and you have this polypeptide chain growing, which the chaperonin will then help fold and make the protein. So that's how proteins are formed, and they are vital for your body. You need proteins. All right, that's our video today.